So I'm from Thermal Powers Lab at Imperial College London, and I'll be talking about a general framework for predicting the temperature dependence of microbial interactions. This is the first section of my PhD work, and I would love any suggestions or any thoughts on this, and especially if you are empirical ecologists, I would love some data to validate these results. So to begin with, I would say no one exists in isolation on this planet, especially for microbes. And in their communities, they compete and cooperate to share resources. Therefore, understanding species interactions is especially important because it determines how individual metabolic activities scale up to shape collective community behaviors. So we study temperature because temperature constrains microbial metabolism and physiological traits. Therefore, it determines how species interact with each other. And microbes constantly experience temperature fluctuations in their natural environment, from minutes to days to seasons, and across different environments. Despite decades of learning on temperature performances on individual species, we still lack the mechanistic understanding of how microbial community dynamics respond to changing temperatures. So I built on the consumer resource theory, this framework built on this consumer resource theory, where interactions emerge from species metabolic traits and resource use. And in this talk, I would like to answer this key question. How does thermal metabolic constraints shape the nature and strength of microbial interactions? This is the MacArthur-style consumer resource model we're using with leakage inside. And for each species, they uptake a certain amount of resource, and part of it is stored as biomass, and then the rest are left system through leakage or resource transformation or respiration. And within the system, there's an input of resource. And with this resource input, species compete and cross-feed, they cooperate with this resource input. And this resource input is actually a function where depending on what kind of system, what kind of environment you are trying to study, it can be constant or teaching or constant environment. It's just adding a term here is output. So within this model, species uptake rates and maintenance respiration rates are temperature dependent. And here is the uptake matrix on the left. So for each species, it sums up to a certain temperature performance curve. And we also have species level leakage as well. So I parameterize this model using unimodal thermal performance curves, where B0 here also contains the scaling effect of cell size in the variation of B0 at pressure. And here E is the activation energy, and T peak is the peak performance temperature. We also included a trade-off between this E and B0, which is activation energy and normalization constant. So with this trade-off, we introduce a trade-off between thermal generalist and thermal specialist. Thermal generalist normally performs quite similar across a wider temperature range, and thermal specialists, they perform relatively better, but at a very narrow temperature range. And this trade-off is quantified by a negative covariance between B0 and E. And these are two very extreme cases where a medium trade-off case, the covariance is 0. And we can have both thermal generalists and thermal specialists exist in the initial community. And this extreme case is a maximum trade-off where the covariance coefficient is -1. And most species within the immutable community would be thermospecialized. But in natural communities, the realistic case will lie between these two extremes. Therefore, we're using an intermediate value of covariance for the results we're generating. So with this consumer resource model, we apply the fast resource equilibration method, which is assuming resource reaches equilibrium faster than consumers. Then we can have the generalized logical theorem model, which is the effect logical theorem model from our model, as the first order Taylor expansion of the consumer dynamic. Here, alpha ij here denotes the impact of species J on species I, which is composed of two compartments, and the first one is the uptake of I on all the resources, and the second one is the impact of other species on equilibrium resource abundance. So the complete equation for this interaction coefficient is right here. And note here, inside this inverse matrix, we have external resource inputs and uptake as well. Here is an example simulation using. There's an example simulation of the temporal dynamics of consumer abundance in Microbe and ELV. We can see the approximation is quite well at around equilibrium. However, there is some deviation in the initial dynamic. So the first result we have is we notice a unimodal temperature performance curve in species interactions, where in species interactions they have higher activation energy and lower T peak, the peak performance temperature. This means interactions are more thermally sensitive than their underlying metabolic traits. This implicates that this increasing temperature, such as in the case of climate warming, interaction networks tend to break before high temperature effects acts on species individual metabolic traits. And we notice species interactions are predominantly negative, and intra-species interactions, the strength of intra-species interactions increase faster than interspecies interactions. The distribution of these interactions shift systematically with temperature, and they are always log normally distributed. We can see the variance of these interactions lies within at intermediate temperatures. Actually, this log normality did not arrive from our thermometabolic constraints. Our thermometabolic constraints actually just enhance this log normality. It emerged from the effects of distributions of uptake rates in the interactions we just showed you. So our interaction results have implications on species diversity and stability. For example, it was pointed out in this paper that the variation of these species interactions actually have an impact on species richness within the community. And we can see the correlation here, where minimal variation corresponds to higher richness in the community. So we have a unit model curve of species richness with temperature. And here this is the ratio between alpha Ji and alpha Ii, which is each interspecies interaction, and their corresponding intraspecies interaction. At intermediate temperatures, the self-regulation tends to be maximized. relative to interspecies interactions. This points to a more diagonally dominant system. Therefore, we get higher stability in the middle. So, in summary, we have these 4 points. Under thermal and metallic constraints, species interactions respond modally with increasing temperature. And effective interactions tend to be low distributed. Interactions are more thermally sensitive than the underlying metallic processes. And at intermediate temperatures, variation of interactions are minimized and self-regulation is maximized. Therefore, we have at these intermediate temperatures, we have higher diversity and more stable systems. That's all.
Any questions?
Thank you. There's something I didn't understand. So here, each species is represented by a temperature response curve. That's the idea.
Yes, there are 2 temperature performance curves. One is for species uptake and one is for respiration.
Okay, and how do you choose the temperatures and the variance of the response?
Oh, that's from empirical data.
Okay.
We use the example set of empirical data to generate the simulation results.
Mm-hmm. Okay.
Yeah, but if you want to use it for different systems, you can just choose your own data set.
Mm-hmm. Thank you.
How does the intermediate temperature relate to the sort of distribution of thermal optima? Like it's intermediate relative to how?
It's paper. You gotta read it.
But yeah, but like, is that more or less where the majority of your thermal optima are, or is where?
It's approximately where the
Okay.
where the crossing is. If you notice in this figure, it's approximately where the crossing is.
Thank you. This is very interesting. In general, I mean, the picture that I have in mind, if you look at, for example, at ocean data, is that diversity is monotonically increasing with temperature. So you have the highest diversity where you have the highest temperature, which is actually around closer to 30 rather than 15.
Patch and how they were adapted to the local environment.
No, I'm talking about like macroecological data set. If you look at Tara Ocean and you look at the relationship between diversity and latitude, diversity peaks at the equator where you have the highest temperature. So I was wondering how you can reconcile these two things.
So actually, how diversity responds to temperature is quite controversial. You can actually see increase, decrease, and all the other patterns. And there was the EMP data. If you look up EMP data, and they have measured all the microbial communities across the world, and they plotted this species richness with temperature, and it's actually a unimodal.
Thank you.
Anybody else?
Hi, thank you. Again, a question on macroecological patterns. So now that your interactions are distributed like a log normal, have you checked like how is the distribution of mean abundances of your species? Because macroecologically we know that microbial communities have a log normal distribution of mean abundances, and it would be cool if with this like a temperature perturbation you could reproduce this observation.
I don't remember results of that, but I do remember that I did check evenness, and it's basically like the richness pattern. If that answers your question.
Okay, but I don't remember the richness pattern. But I mean, we can talk later about that. Ah, I see.
So this pattern look basically exactly like the green one.
Okay. Thank you.
All right, unless there's another question, let's thank the speaker one more time.